Give me 1 second. Sorry, all having a bit of technical difficulties here. Can you all see that? Does that look funky to you? I don't know why it looks funky like that. Okay, is that all right? I'm sorry, I don't know why it won't let me do presenter mode like a normal person, but okay. Hopefully that's all right. Let me know if you have issues seeing anything, and it should also be in your, everything should be in your notes that I've sent already. Okay, awesome. So thanks so much for coming today, everyone. I really appreciate it. Today's lecture is going to be on substitution reactions and biomolecules. But before we start on that, why don't we do a little review of last week? So I have a little practice question for you. Which of the following esters would form from the acid catalyzed reaction of 3-isopropyl pentanoic acid and methanol? And I will give you all a couple minutes to answer the question. You can privately message it to me. You can put it in the chat, whatever you feel comfortable with. Give you a minute. Warm up your brain a little bit. All right, I'm getting some answers. Great, great. Doing a great job. A couple people got their answers in so far. You can keep your answers coming. Okay, awesome. And everybody got the right answer. Also, I have been made aware that Jonathan, sorry not to single you out, you have a really good method for this. Would you like to share how you do these? Sorry, I hate to put you on the spot, but if you have a fantastic trick, I would love to hear it. And you can type in the chat, you don't have to unmute. Okay, you can start by counting the carbons on the reactant. Perfect. And seeing which structure most closely resembles the reactant, i.e. 3-isopropyl pentanoic acid. And then add the methanol, which is +1 carbon. Yeah, that's perfect. That is a great way to look at it. And oftentimes, whatever is most efficient and will get you the correct answer quickly, which, yes, the correct answer is C. Do that method. If you have something that gets you the answer super quickly, super efficiently, do it. Thanks so much, Jonathan. I really appreciate your method. I also have been told that another way to do it is you can also see that there are a certain number of carbons in the name. So pentanoic acid, that's 5 carbons. And if you don't see any others with 5 carbons, they're not the right answer. And then count all the carbons in the reactants. And from there you have your answer. But it's pretty much Jonathan's method. And also, just like last week, I did want to give my little MCAT tip of the week. Given this is my last week, I just wanted to give you a pretty fleshed out tip, but this is my tip for game day. So it's really important to, nobody says this, but plan your snacks in advance. I'm not kidding. Sometimes people forget that it's big test day and they try something new to eat and then they have a little stomach ache on test day, and that is pretty devastating. I mean, it's already hard to focus and take this really difficult exam, and imagine on top of that your stomach hurts. So I'm just giving a few examples of the things that I recommend eating and that I also ate on my test day. So I recommend eating fruits, things that are healthy. Also, fun little quiz question for you. Sucrose is made up of what two monosaccharides? You can put that in the chat as I continue. Try to avoid things that are really heavy and that'll make you sleepy. And especially for those of you with the afternoon exam, I know afternoon can be a really great nap time. So try to avoid, pasta, things that have a lot of carbs in them and that'll make you want to take a nap. Perfect. I'm glad y'all have been paying attention. Yes, perfect. And then avoid foods that you haven't tried before, because like I said, if you have kind of a weaker stomach, that might not end well for you. And then same thing with caffeine. If you haven't experimented with coffee before, don't try it on the day of the MCAT. You never know, and it could make you ill. And so just try to keep it as controlled of an environment as possible. And just, I listed my personal game day meal. Honestly, during the exam, peanut M&Ms were amazing. I also ate them while I was studying, so I kind of had that, put myself in the mental space of knowing all this information. And then, oh, there's a mnemonic that someone uses to remember what makes certain disaccharides. If you want to share that with me, I can share it with the class. So, go grandma, go father son, which is one word, and then go good luck. So go grandma is glucose plus glucose, which makes maltose. And then go father son is glucose plus fructose, which makes sucrose. And then finally, go good luck is glucose plus galactose, which makes lactose, or yeah, lactose. Thanks so much for the tip and the mnemonic. Yes, if you don't mind typing that in the chat for us. Oh, sorry. I do have more tips. So the day before your exam, you will want to study. Try to avoid it. It's oftentimes going to psych you out. And I really just recommend maybe reviewing a couple things that you feel you definitely need a refresher on. At most, maybe look through some passages that you've already done. Review equations that you might forget. Keep it at that. Don't try to learn something new the day before your exam. Again, it probably won't end well. And I really, really recommend just do something you really enjoy and spend time with loved ones. I believe I spent the day before playing Minecraft with my boyfriend, just doing something that I really like, that makes me happy, that doesn't use a lot of brain power, just putting yourself in a good mental space. And then the day of your exam, wake up well before, look in the mirror, say a few positive affirmations, and make sure you feed yourself, because an empty stomach is just as bad as an upset stomach. In fact, you'll probably be having an upset stomach if it's empty. And to get your brain warmed up, try to redo an old Cars passage and maybe a science passage just to make sure your brain's running, get a little warmed up. And then make sure you use the first 10-minute tutorial period and write down your equations, your amino acid chart, all the important things. I believe Todd has probably told you this before, but that 10-minute period is really useful to get down, kind of dump all the information that you know you'll need for the remainder of the test. And then here's some random tips that I've been told and I just personally stuck by to make sure that I didn't get in trouble. So try to avoid wearing a scrunchie. I know most test centers are really strict, and so don't wear it. they'll make you throw it away, and then you won't have your hair tied, um, which could make you uncomfortable. And then also wear a crew neck instead of a hoodie. I don't know if this is true or not, but if you're wearing a hoodie, they might make you take it off. So just be aware, do what you can to avoid making your day more stressful. Interesting. Okay, hoodies will be inspected, but they'll let you wear it. Yeah, you can have a regular hair tie. You just can't have a scrunchie because I guess they think that you'll write on it, which is kind of dumb. Yeah, so it looks like it's dependent on the test center. So honestly, just to avoid putting yourself in more trouble than necessary, I would not personally wear a hoodie. If you have a crew neck, just wear a crew neck. And I know it's really odd with the whole scrunchie situation, but one of my friends did have to throw away her scrunchie, and it's a really big and important exam, and I know people will take extreme measures to get the scores that they want. So I guess it makes sense. But those are my tips for the week. Okay, so just some key takeaways from today. Unlike last week, you will have to know and understand the mechanisms for substitution reactions. However, they are so much simpler and they just occur in 1 or 2 steps, which I'll be going through really quickly. And by the end of the day today, you should be able to know and identify SN1 and SN2 reactions based on information about the mechanism or the reaction, know the conditions necessary for these reactions, know any stereochemical changes if they occur. And then similarly for biomolecules, you just kind of want to know basic information about lipids and nucleic acids and know really well your proteins, your monosaccharides, etc. But we'll go through that. Okay, so before we begin, once again, I want to do a little review of what you should already know. So if everybody would like to put in the chat or send to me privately the stereochemical RNS configurations for these chiral centers. Okay. And I'll give y'all maybe 2 minutes to do that. So there's 4 molecules, so you'll have 4 different answers, 1 for each letter. Okay, I'm getting quite a few answers in. Interesting. Ooh, it looks like I tricked some of y'all. Interesting. Okay. Okay, I will give y'all some more time. Oh, someone said they used Focus to Do and that they really liked it. That is so great. I'm so happy it helped. Okay, I'm getting a lot of answers. It was annoying in a good way. Totally feel you. Okay, getting a lot of answers. I want to give everybody maybe 1 or 2 more minutes just to make sure you're following along. And then we're gonna go into this one actually quite in depth because interesting. Okay. Yeah, so for clarification for C and D, H is not into or out of the page. It's actually in the plane. So the way you would approach that will be a little different. And I will go over all the answers, or let me give you all the answers right now. Okay. So why don't I take my little drawing tool and explain. So before I explain, just in case you needed a little refresher, the way you assign R and S is via the Kohn-Ingold prelog rules of priority. And that name sounds a little scary, but essentially just what it means is the atom with higher atomic number has higher priority. So that means, for example, bromine has a higher priority than carbon. And then if you're comparing isotopes, then the atom with a higher mass has higher priority. So let's say you have deuterium and hydrogen, then deuterium will be higher priority. And then when there's a tie, you will establish priority by proceeding to the next atoms along the chain until you see a difference observed. And multiple bonds are treated as if each bond of the multiple bond is bonded to a unique atom. So let's say you have a carbonyl carbon, that's treated as if you have a carbon bonded to two oxygens. Okay, so the first one, let me draw here. Oh, hold on. Okay, so this is 1, this is 2, this is 3, but because our fourth group is coming out of the page towards you, then you're gonna switch whatever configuration you've assigned. So it looks like R, but it is actually S. Easy enough. This one, 1, 2, 3. So for this one, the hydrogen is going into the page. So we don't need to do any switch of the configuration, and it's going clockwise. So that is R. For these ones, these are kind of tough for me. I am really bad with visual manipulation. So trying to spin things around in my head is quite possibly the worst thing you could ask me to do. So what I do instead is I actually learned this trick from the Berkeley Review book, but I look at the lowest group. So in this case, and for D, it's hydrogen. And I look at whether it is next to group 1 or opposite of group 1. And in this case, group 1 is bromine. So if it is opposite of the first group, the highest priority group, then you'll first find the configuration, assign it a configuration, and reverse the configuration. So I will draw this out. This is 1, this is 2, and this is 3. And as you can see, the hydrogen and the bromine are far apart from each other as compared to the hydrogen and the bromine on D. So normally, this would have been S, but because they're on opposite sides, it is R. So yeah, I got a question on why ethyl is higher priority than methyl. So whenever you look at priority, if you have a tie, so this carbon here, oh, no, let's draw once again. So this carbon here and this carbon here are technically tied. And then, oh wait, that's another way of looking at it. Yeah, so that carbon here is bonded to 2 hydrogens and a carbon. This carbon here is bonded to 3 hydrogens. So this is higher priority. Does that make sense? Thank you. Okay, so as I was saying, we originally assigned S, but because the hydrogen, the 4th group, and the highest priority group are far apart, we designate this now as R. For the next one, let's see, this is 1, this is 2, and this is 3. And I actually might have gotten this wrong. So, oh, I did get this wrong. This should be S, I think. So because the hydrogen and the bromine are on the kind of next to each other, they're neighbors, they're not across from each other, you will take the configuration as is. So in this case, it is S because it's counterclockwise. Sorry, I made a little mistake there. But I hope that makes sense. And then I believe the way Todd taught it was that you point your thumb. I don't know why, sometimes this just didn't work for me, but I'm also really bad at visuals. You point your thumb in the direction of the lowest priority molecule. And then if your fingers curl and they follow the direction of the designation of priority, then that hand is the proper, I guess, designation. So if your right hand follows the correct order of the, let's say, for example, C. If you're, wait, I'm not gonna explain it this way. I changed my mind. Sorry, y'all. I'm confusing myself by trying to do things visually, and I'm just gonna explain what works for me. Yeah, it's a little confusing. Honestly, I stick to my shortcut. Yeah, so let me explain again why C is R instead of S. So the way that I learned it from the book, and this is in my shortcut sheet, is I look at the lowest priority group, which in this case is hydrogen. I look at the highest priority group, which in this case is bromine, and they're apart from each other. They have this distance, as opposed to over here, they're neighbors. Because they are far apart from each other, when you take the priority assigning, you will assign the opposite as what you originally did. So you're gonna have to reverse. And so this is 1, this is 2, and this is 3. So originally this was S, and because of the distance between the hydrogen and the bromine, we have to designate it as R. I don't know exactly why this works. I'm sure there's a visual way to look at it, but it does work. And this only applies when the H is in the plane. And yeah, and some people can visually rotate the substituents. I am, like I said, I am so bad at visualizing things. Yeah, and there's OChem model kits really help. And that trick also works really well. Thank you so much, Stephanie. Yes, the last one should be S. Okay, I'm gonna clear this just to make sure it's not too busy, but I hope that made sense for everyone. So we're gonna move on to SN2 and SN1 reaction mechanisms. Here's a little overview, but we're going, yes, the last one is S. Sorry, I made a little mistake. Okay, and here's a little overview of the 2 reaction mechanisms that we'll be going over in depth today. So SN2 stands for substitution, nucleophilic, and bimolecular. So substitution means that this is a substitution reaction, which, as we learned last week, is when one molecule comes in and substitutes the place of another molecule. Sorry. And nucleophilic means that a nucleophilic compound, compound attacks a substrate. And then finally, the 2 in SN2 refers to the fact that the SN2 reaction has a bimolecular rate determining step. So this means that the rate is dependent on the concentration of 2 reactants. So the SN2 reaction happens in one step. The nucleophile comes and attacks a substrate from behind the leaving group, and this forces the leaving group to leave the substrate, and then a bond is formed. And this is what we call a concerted mechanism. So a concerted mechanism or a concerted reaction is when all the bond breaking and bond making occurs in a singular step. And you can recall that the rate of a reaction depends on the slowest step, as you might have learned in your kinetics lecture. In bimolecular reactions, however, or in bimolecular reactions, because the slow step involves 2 reactants, that means the rate law will include those 2 reactants. And for SN2, there's only 2 reactants. That means the slow step is also the only step, since it all happens at once. And it's also really important to note that one of the identifying features of the SN2 reaction is the attack from the nucleophile on the back side of the molecule opposite of the leaving group. So because this occurs on the back side of the molecule, this causes an inversion of the configuration of the group. So in this case, you have an inversion of chirality as the molecule goes from R configuration to S configuration. And a really nice way to think about this, or helpful for me, I know I said I don't like visuals, but this visual does help me. You can kind of think of this as a really bad quality umbrella, and you have a big gust of wind from underneath it. And that gust of wind is kind of like your backside attack, and it flips the orientation, and now you have an inverted umbrella in kind of a bowl-like shape. So the structure of the alkyl halide in this reaction acts in a pretty similar manner. When the nucleophile attacks from the backside, this inverts the configuration of the molecule, and with that attack, the leaving group leaves at the same time, and this forms a new substituted molecule with an inverted configuration. Does that make sense? Also enjoy my really weird stock images. I don't know how I found them, but they worked perfectly. Awesome. And then now that we have a good understanding of what this mechanism looks like, let's look into what reaction conditions constitute an SN2 reaction. Firstly, given what you know about this reaction mechanism and how it all occurs in one step, meaning that the nucleophile comes in and it just pushes the leaving group off, do you think that the SN2 reaction favors a strong or a weak nucleophile? Okay, I'm getting quite a few answers. Perfect. That's correct. So you need to have a good nucleophile. One that's strong enough to attack the molecule and push the leaving group out while causing an inversion. So you can think of this as a really strong gust of wind. You usually need something strong enough in order to make your umbrella invert like that. So you need a good, strong nucleophile. And just as a little kind of review of last week, can anybody name some strong nucleophiles? I probably gave you the answer after because I didn't want you to have to copy these all down and scramble, but if you can, try not to cheat. Yeah, I'm getting some great answers here. Perfect. Awesome. Okay, yeah, so some good examples of strong nucleophiles are hydroxide, which is honestly one of their favorites, and then amines are great nucleophiles as well. Oftentimes, nucleophiles can be negatively charged because they have the electron density to be able to attack, and highly electronegative atoms are often also good nucleophiles. And there we go. So another important thing, given what you know about the reaction mechanism, do you think that SN2 reactions favor a sterically hindered or unhindered alkyl halide? Meaning, do you think that there should be more substituents or less substituents on the carbon being attacked for an SN2 reaction? Do we want something hindered or do we want something unhindered? Okay, getting a few answers. Unhindered, unhindered. Yeah, unhindered, which is why it attacks from the back. Great job. Perfect, perfect. Okay. Yeah, exactly. So since the SN2 reaction requires that the nucleophile attacks from behind the leaving group, steric hindrance is actually one of the biggest barriers to the SN2 reaction, because as a molecule becomes more sterically hindered, it's more difficult to attack from the back, and then the SN2 reaction becomes less and less favorable. And I have a little chart here showing the actual difference in activation energy needed when you have a more sterically hindered molecule. So as you can see, when you have a primary carbon, the transition state is lower in energy, meaning that this reaction requires less energy to basically proceed to the product. And this leads to a faster reaction rate, a more favorable reaction. And this is why the SN2 reaction prefers an unhindered alkyl halide. I mean, imagine if you had this umbrella with a bunch of things hanging from it. It's harder to make that umbrella invert if you have, I don't know, weights hanging off of it, if it's really bulky, right? So you want something that's unhindered. You want the nucleophile to be able to come in, attack, and just push that leading group out really easy. Okay, so another important thing to consider, the SN2 reaction favors a polar aprotic solvent. Why is that? So hexane is definitely aprotic, but it would make a really terrible solvent because your reactants would not be soluble in hexane. Oftentimes, you need strong aprotic, means that there is no basically ionizable proton, so it's not acidic effectively. It doesn't have any acidic protons. So because your reaction in SN2 often requires a strong nucleophile that's negatively charged, such as hydroxide or methoxide or cyanide, these anions want to be able to dissolve in your solvent more easily. And that's why you can't have a non-polar solvent like hexane, because these polar solvents need to be able to dissolve these salts, since your anionic nucleophiles are often coming in salt form. And the issue for polar protic solvent, why you need a, aprotic, no, but yes. For the issue with polar protic solvents is that the nucleophilic anions will be surrounded by a layer of solvent molecules with hydrogen bonds, and this is called the solvation effect. So what happens is if you have a protic solvent, this is going to form kind of a cage around your nucleophile. It's going to make it a much less effective reactant. And because you have these strong intermolecular forces that you have to break before you can attack, it's much less effective as a nucleophile, and it can't immediately use its electron density or lone pair to attack that carbon and kick off that leaving group. So that's why polar aprotic solvents are preferred for SN2 reactions because they make the reaction most effective. Okay. And here is a little summary of the SN2 reaction. So some features, it favors a really kind of open and less sterically hindered molecule. And in fact, it will not occur if you have a tertiary carbon. So it really prefers primary and methyl halides. And it's also favored with a good nucleophile and a polar aprotic solvent. Some mechanistic features of it is that you have a brief formation of this trigonal bipyramidal transition state. Not super important to know, but just so you can visualize it. And then, of course, important to remember, you form the enantiomer and you have an inversion of chirality, and it's a bimolecular reaction, meaning that second order, both reactants are involved in the reaction rate because they are both a part of the rate determining step, since there's actually only one step, so it's rate determining. And it's a really quick and concerted reaction, meaning that the bond forms and a bond breaks all at the same time. Okay, so also for your consideration, alcohols can undergo substitution reactions. How? And I actually took an AAMC question here and reworded it a little bit, but give it a shot. In order to undergo nucleophilic substitution, an alcohol requires the presence of an acid catalyst. The purpose of the acid catalyst in the reaction is to, and I have all these answers here, and just to help you out a little bit, I gave you the mechanism, but often, I don't think for that question in the AAMC portal, they gave you the mechanism, so you had to kind of know it yourself. And I'm getting some answers. Awesome, awesome. Perfect. Great job, everyone. Yeah, exactly. So the answer is D. You want to create a better leaving group because as we learned last class, hydroxide is a terrible leaving group if you're just kicking off a negative atom. You don't want to do that. But if it gets protonated, water is a fantastic leaving group. So if you do this reaction we can discuss why others are wrong. So produce more dipole interactions in the solvent. As I was saying before, with a polar protic solvent, you don't want that because the intermolecular interactions, that H bonding, etc., that creates a cage around your nucleophile and it makes it really ineffective. With B, you don't really want it. It's not wrong, like it would react with basic impurities, but that doesn't really make sense for the question. It's kind of just an answer to another question maybe, but the purpose of that acid catalyst, as you can see in the reaction mechanism I showed, you can see that that oxygen gets protonated and that makes it a really good leaving group and allows it to go away. And then it prevents the oxidation of the alcohol. That answer doesn't really make sense either because you want to undergo a substitution, not an oxidation reaction. So there's no reason that you would want to prevent oxidation because you're not using anything that would oxidize your alcohol anyway. You're trying to perform a nucleophilic substitution. So that answer just doesn't really make sense either. But yeah, like I said, substitution reactions can occur with alcohols. You just have to make them a good leaving group. And another way for the alcohol to be converted into a better leaving group is to form a tosylate. I don't know if this is something that MCAT tests much, but just for you to be familiar with it, if they ever say or show you this molecule, it makes the alcohol in this really crazy looking mechanism, it makes the alcohol a better leaving group, and it leaves as a tosylate, as I highlighted it on the right side. And just as a kind of little question to poke your mind a bit, why do you think that tosylates make for a good leaving group? Just looking at the molecule, what do you think, what property do you think it might have that makes it a really stable and good leaving group? I see a couple answers. Yeah, exactly. So it's because of resonance. I think size is some of the factor, but the more important factor, and just one of the things that the MCAT loves, is the idea of resonance, because anything that has resonance stabilization is going to be a fantastic leaving group. And as you can see, you have all these pi bonds where the electrons can be delocalized and basically stabilize that negative charge, because the electrons can move around within the molecule. So yeah, just a little kind of thought for you, but not something that's... So alcohols mostly actually undergo SN1, but they can undergo SN2 if it's a primary alcohol. Obviously, it's not going to undergo SN1 because you can't have a stable primary carbocation. But yeah, that's a great question, and we'll be going through that next. So SN1 reactions, a little close look. So as before, the S and N stand for substitution and nucleophilic, but the 1 in SN1 refers to unimolecular. So unimolecular means that the rate of this reaction depends only on the concentration of one reactant. And you'll see why this is. So the SN2 reaction happens in 2 steps. In the first step, the leaving group leaves, and it forms a trigonal planar carbocation intermediate. So in case you forgot your geometry a little bit, no worries at all. Trigonal planar refers to the fact that the carbon is attached to 3 ligands. So, and this is a planar shape. And so trigonal planar, and it's a carbocation intermediate. And this step is the rate limiting step, the carbocation formation. In the second step, the nucleophilic substitution occurs. However, something special about SN1 is that the nucleophile can actually attack on either side of the planar intermediate. So it can either attack from the left side of the molecule or the right side of the molecule, as you can see up close, as I showed. And this results in a racemic product mixture. So to explain what that is a 50-50 or equal ratio mixture of 2 enantiomers, and that results in an optically inactive mixture, meaning that, well, recall that each enantiomer rotates plane polarized light in an equal and opposite direction from its enantiomer. And therefore, when you have the 2 together in equal proportions, that mixture becomes optically inactive, and that's a racemic product mixture. And if you separate the enantiomers, the mixture is said to have been resolved. So the special thing about SN1 is that because the nucleophile can come in from either side of this planar shape, you can have that racemic mixture, that mixture of both enantiomers. And here's the reaction diagram for the free energy of the SN1 reaction. So notice that you can see the 2-step reaction really clearly, and how the rate determining step, which is the biggest hump in this graph, that requires the greatest energy, and that's the formation of the carbocation. And then the nucleophile attacks in the second hump, and this informs the final product. And can anybody tell me the difference between a transition state and an intermediate and how that appears on a reaction coordinate diagram? Sorry, I'm bringing in a lot of different ideas today, but it's good to have a review. So just to repeat the question, can anybody tell me the difference between a transition state versus an intermediate on the reaction coordinate diagram? I kind of helped you a little bit, but just to poke your mind once again. Yeah, exactly. So a transition state is a peak in the graph, and an intermediate is a trough or a dip in the graph. So an intermediate is basically just a short-lived, unstable molecule that's formed in between the reaction when the reactants change into products. And the transition state is just the state before formation of the new molecule. Sorry, I'm losing my voice again. One moment. Okay, and here is a graph showing the differences in the reaction coordinate diagrams for SN1 and SN2. So as you can see, SN1 and SN2 look pretty different. So what causes the leaving group to come off before the nucleophilic attack? I believe it's just the steric pressure of the molecule, which is why SN1, we'll go over this soon, is preferred with bulkier, highly substituted alkyl halides. It kind of just, the sterics of the molecule push the leaving group off. It's not really something that the MCAT could test you on, but basically the leaving group is pushed off by substituents or by steric hindrance. But that's a great question. Thank you for asking. So as you can see, there's a really big difference between the SN2 and SN1 reaction. The SN2 reaction occurs all in one step, whereas the SN1 reaction has the two steps, the first being the formation of the carbocation intermediate. Yeah, so the products in the SN1, it's not drawn to scale. I don't know the actual differences in energy. This is kind of more to visualize. So keep that in mind that it's not, there's no difference in free energy that you need to know for the MCAT. And I don't think I personally learned it at all in my career studying OChem. So yeah, it's kind of just the way I ended up drawing it, unfortunately. But thank you for asking, and glad I could clarify. So as we were talking about this before, based on the mechanism, do you think that the SN1 reaction is favored by a substituted carbon reactant or an unsubstituted carbon reactant? So do you think it would prefer, for example, a tertiary carbon or a primary carbon? Yep. Perfect. And your reasoning is amazing. Exactly. So it prefers a substituted carbon reactant. And why is that? Like people said, carbocation stability. Since the leaving group leaves in the first step of the SN1 reaction to form the carbocation, it is really important that you have a stable carbocation in order for the nucleophile to attack. And carbocation stability is actually the greatest barrier to the SN1 reaction. So compared to SN2, where the barrier is sterics, since the the nucleophile has to come in and attack from behind for SN2, SN1 reaction, on the other hand, the greatest barrier is the stability of the carbocation. So here's a little chart showing carbocation stability. Carbocation stability is directly related to the number of carbons attached to the carbon. And thus, SN1 reactions are highly favored with tertiary alkyl halides. And in fact, the SN1 reaction pretty much does not occur with primary and methyl halides because those carbocations are extremely, extremely unstable and you just simply do not want to form them. Now, for solvents, the SN1 reaction favors a polar protic solvent. And why is that, you might ask? Stability. So in the SN1 reaction, the leaving group leaves and a carbocation forms. And this is also the rate determining step. A polar protic solvent, such as water or maybe methanol or ethanol, is able to form hydrogen bonds with not only the leaving group, but also the carbocation. So the oxygen, as you can see in the little drawing I have there, its negative electron density interacts with the positive electron density of that carbocation, and that stabilizes it because, as you know, in the second step of the reaction, you need that nucleophile to come in, and before it can do that, you want it to be a stable carbocation and be there as long as possible for that nucleophile to come in and attack. So anything that can stabilize the carbocation will speed up this reaction and make it more favorable. And this is why you want a polar protic solvent. And it's actually also very common that the polar protic solvents can serve as a nucleophile themselves for SN1 reactions. And so sometimes you'll hear SN1 reactions being referred to as solvolysis, which essentially just means that the solvent is acting as the nucleophile. So just to kind of summarize what we've gone over with SN1, the favored electrophiles are going to be the tertiary electrophiles and then the secondary, and then you pretty much do not want a primary electrophile. It's not going to happen. Just don't think about it. It won't happen with SN1. And they are favored with good leaving groups to form a carbocation easily. And like I said, sterics will push that leaving group off, as someone asked before. And it is favored with a polar protic solvent because, like I said, you need something that will stabilize that carbocation and stabilize the nucleophile for it to attack. And then you have formation of a trigonal planar or 3-ligand transition state, and this is your rate determining step. You have your solvent, which can act as a nucleophile, and some product features is that you form a racemic mixture since the nucleophile can attack on either side, and there's a possibility for rearrangement, which I'll go over in a second. And it's a unimolecular, which means that the rate only considers the concentration of the electrophile because it really only matters how many carbocation intermediates you can form in order to proceed, since that step is the rate determining step. And it is a slower two-step reaction. So just a quick note on rearrangement. I haven't seen any testing materials about this for the MCAT, but it's just something that you might want to know. Essentially, if there's a possibility for a tertiary carbocation to form, it will. And this will occur via a hydride shift or a methyl shift. And so the rearrangement occurs via the mechanism that I've shown, and this creates a more stable carbocation, and then the nucleophile attacks. And this forms a different product than you might have expected from the beginning. And essentially all this has to do with carbocation stability, but again, I'm not sure how heavily this is tested on the MCAT. I haven't seen any resources at all include this or mention this, so it's kind of just for your own knowledge and to help make sense of the fact that rearrangement does occur and how it occurs. And finally, here's a little summary chart of SN1 and SN2 reactions, their key features, stereochemical changes, if any, the electrophile that's preferred. Oops, I messed up that a little bit. But you can fill that out on your own. I guess I meant to put that for the electrophile row, it should be for SN2 primary, and then is most preferred, and then for SN1 is tertiary most preferred. Okay, so anything that stabilizes a carbocation is characteristic of an SN1 reaction. Yes, exactly, because that is the rate determining step of your reaction, that first step where the leaving group leaves and forms a carbocation. So things that make that first reaction occur quicker will make the reaction more favorable. And it forms quicker if it's more stable. So, yep, that's a little summary of our SN1 and SN2 reactions. I did make a little oopsie on the electrophile section, but that is also in the other tables that I have, so you can just refer to those or change it yourself. But now we're going to move to some practice problems. So these are something that Todd really enjoyed, and honestly, it helped me a lot for the MCAT. So I'm gonna give you a couple of these, and it's gonna be really weird, but I promise the skill behind it is extremely helpful. And just kind of a note about this, you are going to come across questions where you have absolutely no clue what they are asking. I remember just feeling so confused sometimes, and especially when I first started the exam, I remember physically just sweating because the first passage they gave me for Chem Phys was disgustingly difficult. And I was sweating. I was like, I don't know what they were asking for any of these questions. I'm going to move on. And I came back and I used the skill and it helped me a lot. So we're going to go through a couple of blah, blah, blahs, as Todd calls them. So the first blah, blah, blah. What do you think? Give us your answer and feel free to give your reasoning. I'll give you a couple minutes. I know these are super weird at first, but it's such a useful tool when you're taking that exam, because I guarantee you will come across questions that you just do not know what is going on. Okay, I'm getting a couple answers and then I'll give everyone a little bit more time. Okay, so the answer is indeed D. So let's go through that. If you look at each of the answers, you'll notice that they all refer to either an SN1 or an SN2 reaction. So we're going to go through each of them individually. The first one, the solvent can act as a nucleophile. We learned the term for this before. It is solvolysis for SN1. The rate does not change as a nucleophile is added. So recall that SN2 means bimolecular and SN1 means unimolecular. And in SN1 reactions, the unimolecular rate determining step is that formation of the carbocation, and therefore the rate is only dependent on how much electrophile you have, how much carbocation trigonal planar intermediate you can form. And so this applies to SN1, because if you add more nucleophile to your reaction, it's not going to change the rate at all. It's not included in the rate determining step. And then C, optical activity is lost over the course of the reaction. So this is also SN1 because if you can recall, in an SN1 reaction, since you have a trigonal planar intermediate and the nucleophile can attack on either side, you have a formation of a racemic mixture or a mixture of two enantiomers that rotate polarized light in opposite directions, and therefore they cancel each other out. And as the reaction proceeds, you'll lose the optical activity as the, yes, one of these things is not like the other, as someone just said. And that one of these things that is not like the other is this. And this refers to SN2 because you have a change in the sign of the specific rotation, which is indicative of the enantiomer forming. Okay, great job, great job. Give me 1 second. I lost my little. Okay. Our next one, wee woo, blah, 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 except blah, blah, blah. Give you a couple minutes to figure out the answer here. The MCAT loves except questions, so get used to answering them. Yep. Yep. Getting some great answers. Awesome. Okay. So I'll give it maybe 1 more minute for everyone to get their answers in after thinking about this blah blah. Okay, looks like we have all of our answers, and yes, your answer is D. So we'll go through this one once more. So A, it is preferred with a primary electrophile. This is very much SN2. Like we said, a carbocation that's primary is super, super unstable. That is not going to want to form. SN2 very, very much prefers tertiary and at times secondary, but mostly tertiary electrophiles. And the second one, B, it has no intermediate in its mechanism. As we know, the rate determining step of an SN1 reaction is the formation of that trigonal planar carbocation intermediate. So that is also SN2. And then it is preferred in a polar aprotic solvent. We know this is also SN2 because we don't want the nucleophile to be essentially distracted, caged in, bonding with anything else. We want it to just go straight to that nucleophile and kick that leaving group out. And this, of course, hydride and alkyl shifts, as we mentioned before, that has to do with rearrangement, and rearrangement is an SN1 phenomenon. Awesome. Right. Great job. And now some real practice problems. So while performing a reaction to form a pharmaceutical compound, you find that the reaction is initiated when a carbocation forms. Which of the following is likely true? And then we have 1, 2, and 3 here, and all your answers. And I'll give you a couple minutes for this one. Okay, maybe a few more minutes. Getting a couple answers. So we'll give everyone a little more time, plenty of time, so no rush. Okay. Awesome. Great. Okay. So I have a couple of answers. I'm going to go ahead and the answer is D. So we can go through this one as well, just in case we need extra explanation. So number one, concentration of the nucleophilic halide is the main determinant of reaction rate. So this is referring to the reactant that looks like, for example, this, or let me give it an actual. So this is SN1. As we know, the reaction rate is determined pretty much, it's just exclusively by the concentration of your electrophile, which is the, oh wait, I think I messed this up. It should say alkyl halide, which is why I think there's a bit of confusion. Okay, this should say alkyl halide. But the answer is D, if that says alkyl halide. The second one, the carbocation forms when the hydroxide forces the bromide off the off the carbon. This is an SN2 mechanism move in which the reaction rate is determined by how much of the electrophile you have. And your electrophile is what forms that carbocation intermediate, which is why it determines the reaction rate. Sorry for confusing that, but essentially how much electrophile you have determines how much carbocation intermediate you can form. Yeah, the alkyl halide is the electrophile. It shouldn't say nucleophilic halide. Yeah, so an alkyl halide is just anything that, any carbon compound that has a halogen group attached to the carbon, so like that or like that. But for an SN1 reaction, you wouldn't have this. And then the third one, the carbocation is planar. As we mentioned many times before, it's going to form that planar, trigonal planar intermediate, and that nucleophile can come in from the top or the bottom or the left or the right, however you want to envision that. Yes, exactly. The alkyl halide forms the electrophile after the leading group leaves. So I can just draw that real quick. Let's say you have this and the bromine leaves, and that forms your trigonal planar carbocation. So your answer is D because these are referring to SN1. And if you see in the base of this question, it says you find that the reaction is initiated when a carbocation forms, which is a signature of SN1. So immediately you know, okay, I have to find the answers that have to do with SN1, which are 1 and 3, and that gives you your answer D. I'm going to take this off. Okay, number 2. What is the most likely stereochemical configuration of the products resulting from the following reaction? So I'll give that a couple minutes. Okay, I have a couple answers in. I'll give it 1 or 2 more minutes. Perfect, perfect. Awesome. Exactly. So your answer is A. So let's look at why that is. Oh, yes, I can go back to the last slide. The answer is D, and also something important to, sorry, I cleared all my drawings, but this should be alkyl halide and not nucleophilic halide. So let me write that real quick. Sorry, my trackpad drawing skills are horrible. Um, no worries, no worries. If okay, you're very welcome. Let me go back to this one. Okay, so I know it looks a little confusing, but let's see why the answer is A. What is something you notice, especially about this right here, that tells you what your answer is? What do you notice about this molecule? Exactly, carbocation. And also, what is the property of this molecule, molecule 1? That kind of hints towards your answer. And you look at it and you kind of think, interesting. Beyond just looking at step 2, because let's say they didn't have step 2, if you look at that first molecule, what about it? Exactly, it's tertiary. You have 3 things bonded to that bromine, and this is pretty bulky. I mean, it's a big molecule. That is not going to want to be attacked by a nucleophile. Um, and there's very clearly a carbocation in the second step, which is very, very much SN1. And so when you have an SN1 reaction, that SN1 intermediate is the carbocation that's planar, and the nucleophile can attack on either side, and this forms a racemic mixture. So let me go to the next slide real quick. The answer is A. Let me know if you have any questions. Like I said, we have plenty of time, so feel free to make me go back to any other slide if you need further clarification. Okay, practice problem 3. A pharmacologist modifies the above reaction by making changes to the reactant, replacing a hydrogen attached to the central atom with an ethyl group. The new reactant formed now has 1 methyl group, 1 ethyl group, and 1 hydrogen attached to the central carbon. How will this modification affect the reaction? Also, I realized that I messed up. This should be, um, the product should have an ethyl group. Sorry about that. Okay, I will give it a few more minutes. I have a couple answers in. So I know it's saying a lot, and the MCAT really likes to speak a lot, but say very little. But let's break it down what they're asking and what they're saying. So they are taking 1 hydrogen group and they are replacing it with an ethyl group. So let's just draw that here. How does that change the reaction that is occurring, which based on the original molecule and what that used to look like, and let me redraw it so it's a little easier to see. Do we think that speeds the reaction up or slows the reaction down based on how the molecule has changed? And I can draw the new molecule here. what do we think about the reaction speed? Will it go faster or slower? Yeah, exactly. This reaction will go slower because um, if you haven't been able to see already, this is an SN1 reaction. Originally you had um, a primary alkyl halide, and now you have a secondary alkyl halide. And if you go from primary to secondary alkyl halide, it is, so both of the reactions were SN2 because as you can see, there's no carbocation formed and there's no mention of carbocation. It is an did I say SN1? Sorry, SN2 reaction. And you've gone from a primary carbon to a secondary carbon. This is going to slow down the reaction because now you have more things blocking the way of your nucleophile and making it more difficult to push that leaving group out. So yes, steric hindrance is increased, but the reaction proceeds slowly, more slowly. So it's an easy mistake to only read one half of the answer, and CARS loves doing this. So it's a really good skill to read the question and the answers thoroughly, because if you only read the first half of the question, oftentimes they'll have the rest of the answer, or one half of the answer, sorry. The rest of the answer might be wrong. And so this is your correct answer actually, because your activation energy is higher. Since this reaction is less favorable, it requires more energy to proceed. And as a result, the reaction proceeds more slowly. It's more difficult for that nucleophile to come in and attack. And therefore the reaction, no, it's an SN2 still. Because there's no mention of a carbocation and there's no mention of any like leaving group leaving. It's going to be the same reaction as in the first step, as this pharmacologist did. He's basically saying what happens when you have this primary alkyl halide and now you've changed it into a secondary alkyl halide. How is that going to affect the original reaction? So there's been no change to the reaction, if that makes sense. But yeah, like I said, make sure you read the whole answer, since the MCAT loves to trick you, unfortunately. And for, I would say, CARS, this is so common. They will give you a half right, half wrong answer. Yes, so that's correct. So the activation energy is higher. And this is in a previous slide when I showed you what happens actually when you change, or when you go from primary to secondary alkyl halide. The activation energy is higher because it requires more energy for this reaction to occur. It's less favorable because the nucleophile encounters more steric hindrance when it is trying to attack the carbon from the backside, because there's just more there and it's difficult for the nucleophile to access the leaving group from the back. And so that requires more energy to occur, and the reaction is less favorable. The reaction occurs more slowly because, like I said, it's difficult for that nucleophile to access the carbon. So just to reiterate, SN1, the biggest kind of barrier to an SN2. The biggest barrier to an SN2 reaction occurring is steric hindrance. And that's why primary alkyl halides are so much more preferred than secondary alkyl halides. But they can still occur with the secondary alkyl halide. It'll just be slower. You're welcome. Okay. And I know this might be your third biomolecules lecture, so I'm not sure what exactly has been gone over already. So I'm just going to kind of do an overview of biomolecules and kind of let you know what I think is important to know, and based on my experience studying for the MCAT and taking the exam. So with proteins, what to focus on? Amino acids. I know you've definitely had this lecture already, and Todd has probably said this a million times, but absolutely know the structure extremely well, know your 1 and 3 letter abbreviations extremely well, know the properties of your ionizable amino acids and your acidic and basic, etc. amino acids. Um, just know everything that you possibly can about your amino acids. Make flashcards, use Quizlet, anything, just absolutely know them. I mean, they show up in virtually, I guess, 2 sections. So they show up in both the biology and biochem section, and they also show up in organic chemistry, or sorry, the chem and ochem section in physics. So just know them. It's easy to kind of procrastinate, but start. Todd's trick of calculating PI quickly. I don't have that off the top of my head, but I will be sharing all my cheat sheets with you all, which should have all the shortcuts and tricks. But yeah, that'll be coming in an email soon. Sorry, I'm still putting together this folder. And then know the chart. I believe he should have given you this chart in the amino acids and proteins lecture, but if not, I have it here. I copied this chart down during that 10-minute tutorial period. It was really helpful in just knowing all the, okay, he gives it like every other week in lectures. Perfect, yeah. This chart, super helpful. Know it, copy it, practice copying it. It's really important. The MCAT loves PI, they love PKs, they love electrophoresis, all this stuff. They really like it. I'm not sure why, but it always shows up on the MCAT. So I'm glad it's seared in your heads. Just make sure you know it. And of course, GluAsp and his lies are basic. I'm sure you've heard those before, but just in case you needed those mnemonics, I always remember acidic as glue, asp, and then his lies are basic, or his lies are basic. And then also know your structure. This is on the next slide, but know the different levels of structure. Oh, sister, yes, I did forget those too. So know your protein structure. Just as a refresher, your primary structure is your sequence of amino acids. So let's say you have a chain of amino acids that's A, D, E, G. That's your order of amino acids is your primary structure. Your secondary structure is the hydrogen bonds between the backbone atoms. So this includes your alpha helix and beta plated sheets, which form when you have the carbonyl oxygen and the nitrogens bonding in the backbones of your amino acids. And proline and glycine can play a role in your secondary structure. And also something important to note is that detergents such as urea can break down secondary structure. So sometimes the MCAT will mention urea. So that's something just to know. And then tertiary structure. So tertiary structure is interactions between side chains within a polypeptide. So these can include non-covalent interactions, which means any interaction that is electrostatic or hydrophilic or hydrophobic. And it also can include covalent interactions, which refers to formation of disulfide bridges. And this is when you have, just as a little refresher, a cysteine and another cysteine, and they are oxidized to create a bridge between the two sulfurs called a disulfide bridge. And finally, also just to mention, these covalent bonds are a lot more stable. So you need something called a reducing agent, which, as we learned last week, is something that causes reductions to be able to break down the disulfide bridges. So reducing agents are different than detergents. Detergents are what break down the other interactions, like the secondary interactions, but reducing agents are needed to break disulfide bridges. And then finally, we have tertiary protein structure, which is the side chain interactions between different polypeptides. So as an example, hemoglobin contains 2 pairs of different proteins, and they're designated as alpha and beta. You don't need to know that, but it's just an example to keep in mind as an example of quaternary, not tertiary, quaternary structure. And then carbohydrates, what to focus on. Definitely understand how to read Fisher and Haworth projections, being able to determine D and L from these structures, the anomeric carbon, anomers, epimers, just know how to look at these structures and be able to determine whether also you have an alpha or a beta bond, and feel free to look back on your old notes if you need a refresher on that, and I'll be sharing my cheat sheets regarding that soon. Mono and disaccharides, know their structures, know the common disaccharides, use your hands. I believe you should have had this lecture already, but if you haven't, it's so important. I think I got maybe 2 questions, but still those are easy points on monosaccharides, and I had to know their structures, and I'm like flipping myself off in the middle of my exam just to make sure I know glucose. So like I said, F-glucose, and then there's mannose with the gun signs, and then galactose is the C4 epimer glucose. And reducing sugars, refer to the last lecture, but these are sugars that contain a hemiacetal, which is basically a carbon that's bonded to an hydroxyl group, which allows it to be oxidized. And Benedict's and Tollin's reagent are just 2 basically solutions that help you determine if you have a reducing sugar in a compound. I believe they change colors due to some reaction that occurs. And then know your biochemical pathways. I personally did not see this on my exam in terms of actually knowing, for example, aspartate, alpha ketoglutarate, specific orders of the Krebs cycle or glycolysis. I didn't really have that, but the MCAT really likes regulation of these pathways. They really like hormones. They love, love, love insulin, which makes sense, but just know them in case it's easy to memorize them. It's kind of just brute memorization. You put in the time and you should be able to memorize these. And it's better to know way more than you need to than have deficits in your knowledge. Okay, for lipids, what to focus on. So saturated versus unsaturated. So can anybody tell me saturated fats are solids or liquids at room temperature? Yep. So saturated fats are solids at room temperature. And I know it can be kind of confusing, but saturated fats refer to the fact that they are saturated with hydrogens. And because they have basically the ability to pack really tightly, they can form solids at room temperature. Whereas unsaturated fats have double bonds, which create kinks, and it's more difficult to pack them because double bonds don't really bend well. They don't turn and twist and rotate. So unsaturated fats, which are fats with double bonds, are liquids at room temperature. And then understand triglycerides. Triglycerides are actually created via esterification, which we learned last week. And their functions, they're very efficient storage, insulation, packing, and then phospholipids, know that they're amphipathic. This is different from amphoteric. So amphoteric is a molecule that can be either an acid or a base, whereas amphipathic refers to the fact that it has polar and nonpolar character. So what makes phospholipids so important in our bodies and in biology is that they have both that polar head and that nonpolar chain, and they can form, membranes, micelles, etc. And then no terpenes. These are used to make waxes, but they can also be cyclized to make steroids, which are really important for our body. And steroids, their functions are basically to regulate the stability and the fluidity of the membrane. So when it's cold, the membrane wants to become rigid, and cholesterol can separate phospholipids and prevent the membrane from becoming too rigid. And then when it's too hot, cholesterol can decrease the fluidity and prevent the membrane from becoming a little too fluid. And they're important for bile salt synthesis, other things. And then, like I said, understand why phospholipids are important, and that's due to their amphipathic nature, so they can form the phospholipid bilayer and micelles. And like I said, since water is polar, that hydrophobic region will aggregate while the hydrophilic region interacts with water around it, and this allows for micelles to form. And then nucleic acids, what you should focus on. So, I don't think I said anything about sphingolipids, but just know the structure. I have never seen a question on a sphingolipid, but just be able to familiarize yourself with what the structure looks like. I don't think they're particularly important. And in my experience, I haven't seen any questions asked about them. And then nucleic acids, what to focus on. So be able to first understand what makes up a nucleotide. But something that I don't think is emphasized enough is actually knowing the structure of the nitrogenous bases. I had a lot of practice questions with these, and I think I had maybe 1 or 2 questions on the MCAT asking about the structure of the bases. So you should be able to identify them by looking at them, which I know can be tough, but the more you practice and the more you kind of flashcard them, the easier it is to remember what they look like and be able to kind of create tricks to remember what's what. I've also had people come to me wishing that they had known to study the structure of the bases. So you heard it here, know the bases, know what they look like, and also understand the hydrogen bonds between them. I've had, I don't remember if it was on my MCAT or practice materials, questions about how many hydrogen bonds each base donates or accepts and where. And if you do a little Google search, there's a really nice photo showing where in the molecules the H bonds occur and how many are donated and accepted. I wish I had put that on there, but I would just look that up just for reference, because I also think that's important to know. And then, of course, understand your lab techniques. So PCR, DNA sequencing, gel electrophoresis, super important. But yeah, that is all for today. I believe we're going to end a little early unless anybody has any questions. Oh my gosh, I didn't realize my mic was muted. I don't know how that happened, but thank you so much, everyone. I will be getting that cheat sheet folder to y'all soon. I'm not sure if everybody wants my notes, because I did take notes on most of the chapters in the books as well. I guess I could include them. Yeah, so I will try to get all that to y'all soon. Just bear with me because it is almost finals week. And oh, yeah, so I will send you my email and you can just email me once again and say, hey, TBR has the wrong email address. Also, it might be helpful to email Josh that they have the wrong email because for a long time they were sending my dad emails. So it's an easy fix. Thank you so much. And yeah, I'll be getting that cheat sheet folder to y'all as soon as I can. There's a lot of notes I have, so it'll take me a while to just put it all in one folder, but it should be okay. Yeah, if no one has any further questions, class is dismissed. Yeah, I can re-upload the slides. You're very welcome. Also, this is my last lecture. So thanks so much everyone for being a great audience and listening to me. I don't know if you heard, but Berkeley Review is not taking another class in the spring, so I really only got to teach these 2 lectures, but it was a great time teaching you all, and I'm happy to spread the knowledge. Yes, so after I get through finals, I would love to open office hours. I don't know if Todd will or Josh will let me because apparently I'm supposed to be done by mid-December, but if I am allowed to, I will open up office hours because I'll have a lot of free time once I'm done with my semester. But yeah, I would love to do office hours. It's just a matter of I have been so swamped this semester coming back from COVID and just hitting the ground running. But to answer your question, yes. Yeah, of course. I answer my emails very frequently. So if you have any questions just like about your personal MCAT experience and would like assistance, I'm very happy to help. Just shoot me an email. I'll drop my email one more time for anybody who wants it. But yeah, I'm happy to help. So if you have any questions, MCAT or OChem or otherwise, just let me know. Yeah, does anybody have any other questions? Otherwise, I will close Zoom. Maybe I'll stay for like 5 more minutes. Thank you, you too.